Good morning, everyone. Let's start po. Okay. Good morning, everyone, and welcome to the exit report presentation of Balik Scientist Awardee Dr. Jhalique Jane Fojas. I am Cherilyn Chow, your host for this morning event. This is made possible through the support of the Department of Science and Technology, Philippine Council for Health, Research and Development, Balik Scientist Program. Also, this event is live streamed through the UP Diliman Department of Chemical Engineering Facebook page. Feel free to share the live stream via Facebook. We would like to acknowledge the University of the Philippines University Computer Center for arranging this Zoom webinar. Dr. Fojas was hosted during her Balik Scientist stint by the University of the Philippines Diliman and the University of the Philippines Los Baños. In her exit presentation today, Dr. Fojas will be giving a lecture on microfluidics, future of precision diagnostics. After the lecture, we will have a question and answer portion. You may start writing your questions on the Q&A box here on Zoom. Those watching live on Facebook may also write your questions through the comment section. We hope to entertain all questions if time permits. Now, to formally open this event, let us hear from the Vice Chancellor of Research and Development at the host university, the University of the Philippines, Diliman. Please welcome the Vice Chancellor for Research and Development, Dr. Gonzalo Campoamor II.
Hello, magandang umaga sa inyong lahat. Sa ngalan ng ating Chancellor Fidel Nemenzo, ay wini-welcome ko kayong lahat para dito sa exit report presentation ng ating Balik Scientist Awardee, si Dr. Jhalique Jane Fojas. At sa ngalan din ng buong UP Diliman at ni Chancellor Nemenzo, ay kinocongratulate natin si Dr. Jhalique Jane Fojas para sa kanyang makabuluhan, napapanahon at masinop na pananaliksik na pinamagatang Microfluidics, Future of Precision Diagnostics. Na palay ko ito'y kailangan nating teknolohiya, pananaliksik para sa Microfluidics technology, lalo may kinalaman ito sa diagnostics, treatment at saka management ng mga sakit, no? Lalo pat nasa loob tayo ng pandemic ngayon. Gayon din, sa ngalan ng UP Diliman at Chancellor ay kinocongratulate natin, bukod kay Dr. Fojas, ang UP Diliman Department of Chemical Engineering at nagpapasalamat din tayo sa lahat ng naging bahagi at tumulong dito sa proyektong ito, particular ang DOST PCHRD, kasama natin ng mga representative mula sa PCHRD ngayon. Pakipaabot na lang ang aming pasasalamat kay Director Jaime Montoya at kasama rin natin ang partner ng UP Diliman, ang UP Los Baños sa pamamagitan ni Vice Chancellor Rex de Mafeles na sa maikling panahon, sir, ay nagkasama tayo sa AAC at masaya po ako na muli tayo magkakasama ngayong araw. Sa lahat po ng mga nakikinig o nanonood sa Facebook Live, ay pakinggan natin mabuti ang mga sasabihin, ang report ni Dr. Fojas at gaya ng sabi kanina ng ating host, ay maaari kayong magtanong pamaya-maya at sana ay maging productive ang ating umaga. Marami pong salamat and congratulations again, Dr. Fojas.
Maraming salamat po, Vice Chancellor Campoamor, sa inyong napakagandang mensahe. A round of applause, please, for Vice Chancellor Campoamor. And now, to introduce our main speaker, please welcome Professor Rizalinda De Leon, who hosted our speaker during her Balik Scientist stint at the UP Diliman Department of Chemical Engineering late last year.
Thank you, Ma'am Cherry, and good morning, everyone. Good morning, Vice Chancellor Campoamor. And before I introduce our speaker, I would like to acknowledge the presence also of our guests from DOST, Ma'am J. Calo of the DOST Office of the Undersecretary for R&D, and Ma'am Laila Labonite, and our colleagues from the DOST PCHRD. I would also like to acknowledge the presence of Vice Chancellor for Extension Research, Dr. Rex Demafelis from UPLB. Our Balik scientist is an expert in the fields of biomedical and chemical engineering, and biomaterials and biomedical devices, microfluidics and organ on a chip, nanoscience and nanotechnology, neuroengineering, neurophysics, medical sciences, and nanomedicine. She is currently a research fellow at the Harvard Medical School. She finished her Bachelor of Science in Chemical Engineering from the University of the Philippines, Los Baños, and her Master of Science in Chemical Engineering, her Master of Science in Applied Mathematics, and Doctor of Philosophy in Chemical Engineering from the University of the Philippines, Diliman. I am proud to say that she was my thesis and dissertation advisor. In 2015, she earned or finished her Master of Research in Physical Sciences at Cambridge University, and right after that, started her PhD in physics at the same prestigious university, while also obtaining a certificate in psychology from Oxford University. As of 2019, she has finished all her requirements for her doctoral degree at Cambridge and is waiting for the formal conferment, which has been suspended due to the pandemic. While waiting for that, she started her Master of Medical Sciences in Clinical Investigation, which she finished just last month, and is currently continuing her Doctor of Medicine studies at Harvard Medical School. Prior to 2019, she has worked as instructor at UP Los Baños, as a research associate at Harvard MIT, and teaching supervisor at Cambridge University. She has received fellowship grants from the American Association of University Women International Fellowship, the Harvard Global Health Delivery, and Schlumberger Research Fellowship. Ladies and gentlemen, please welcome DOST PCHRD Balik Scientist, Dr. Jhalique Jane Fojas.
Magandang umaga po sa lahat. Highlight. Let me just share my screen. Magandang umaga. First, I would like to thank the DOST, PCHRD, and UP for arranging and making this lecture possible. In this talk, we will basically explore, albeit concisely, recent microfluidic platforms and micro-nanotechnology designed to facilitate disease diagnostics, which is very relevant with the current pandemic situation, management and detection of agents and disease, and treatment development such as drug screening and drug delivery, and how this microfluidic technology can be translated in the Philippine setting. Because this is an exit lecture, I will also be going through the things that a short-term BSP program such as this one can achieve in a few months in terms of establishment and incorporation of microfluidics technology in academic, industrial, and biotechnological research in the country, with, of course, emphasis on health applications. So point of care testing, or what we call POCT, allows clinicians and clinical scientists like me to detect and diagnose diseases at or near the patient side, or on-site, called on-site, relatively in a faster and portable than conventional lab-based testing. So we've seen this so much about this, we've heard so much about this during the pandemic. So the importance of POCT, or point of care testing, is considerably amplified with the current situation of COVID-19 and also other viral infections, not just affecting humans, but also livestock or animals. Although numerous point-of-care tests and diagnostic devices are available in the market, including but not limited to, let's say, your typical glucose monitoring, your pregnancy test and infertility testing, or other common infectious disease testing, the other markers for disease detection and also drug screening, from drug discovery to drug development, is quite limited in the Philippine setting. So integrating microfluidics in point of care testing would allow certain fluid manipulation and detection in a singular device with minimal sample requirements. And this is quite economical and highly portable and very much applicable in a country such as the Philippines. So during my Balik Scientist program, I've dealt with giving lectures on not just fundamental research regarding microfluidics, but as well as applications of microfluidics, training people, researchers and scientists how to use and fabricate microfluidics for simple and complex applications. So if you've attended some of the webinars or the lecture series, you would have a basic idea that the microfluidic application range from fundamental research such as studying the phenology or phenomenon of a certain event, such as in here, you have here the diffusion, for example, or you could study as well as the drug diffusion on the left side, or you could do interface research, which is also fundamental research of that you could utilize microfluidics platforms for, so solid and liquid interactions. So it varies really. So microfluidic technology varies from simulations of simple phenomena to complex systems to eventually developing and manufacturing actual devices that could be used for hopefully point of care medical applications. So the target for a majority of the research works that I've been doing is on biomedical and health applications. So in the 3 to 4 months that I've been giving lectures and developing proposals and establishing microfluidics technology in the Philippines, I have incorporated both the fundamental, applied, and marketable technology seminars in the process, and also have started working on several research applications that are very much relevant in the country. So in the next slide, you'll see here some of the applied applications that could be translated in the Philippines. So you could have here the detection for bacteria in beef. So that's your center figure. So this technology has been developed by scientists in the US and Japan, wherein they could detect bacterial contamination in beef. So this type of microfluidics application is very much relevant in the country because we have a lot of livestock. We have a lot of not just cows, but also pigs, swines, which I believe a few months ago have been highly infected by African swine fever. So all these things could also be detected using a microfluidic device. Additionally, we could do environmental applications for microfluidics. So because, the relatively small size and precise flow control, it will enable us to do any sort of in situ process to visualize it. And then increasingly, we can do much more applications in the environmental science and engineering as well. So you can do various sensing for water contaminants or like heavy metals and even organic pollutants. We can do that using microfluidics as well. We could also detect microorganisms in the environment, let's say bacterial or viral contamination in water. And at the same time, we could do that in solid or in organic materials, like the one that I mentioned for agricultural or veterinary applications if the meat is contaminated or polluted. And we can also do a lot of research when it comes to, let's say, biofilm formation. So those could be also used for encapsulations of important compounds or molecules. Now, a majority of the applications of microfluidics have been geared towards medical and biomedical technology or biomedical advancement in the last couple of months in particular because of the pandemic. And that's what's something that is the final goal in the Philippines really, is to be able to develop microfluidics to the extent that we could actually use that in health application, diagnostics, and point of care platforms. So in the next slide, I'd just like to show you how important microfluidic is currently. So a lot of studies regarding marketability of the technology have shown how much we could actually utilize microfluidics for a lot of lab testing technologies such as microarrays and ELISA, all this, and even RT-PCR. So RT-PCR is currently what we've been using to detect COVID-19 in samples, in swab samples in humans. But all this could also be designed using microfluidics. And that would mean that it is highly portable. It is more economical because we won't be using a lot of expensive reagents which we have to import in other countries. So this type of technology, if translated in the Philippine setting, or if developed in the Philippines, could help us make economical and portable devices in terms of diagnostics. And also the same technology could be used for drug discovery and development. So a lot of works on drug screening using microfluidics, being a high throughput system, has been utilized or has been explored the last couple of years in various countries. Additionally, like I mentioned a while ago, we wanted to exploit microfluidic technology and even nanotechnology in developing clinically relevant devices, so clinically translated devices such as diagnostic devices. So in this particular slide, you can see here something that we've manufactured through UP Diliman, Philippine Science High School, and Ateneo de Manila. So this is actually a lung-on-a-chip device. This was first developed in Weiss Institute at Harvard. I believe I was there when I was doing my sandwich PhD program when they started working on this. And you can see that with the current equipment that we have in the Philippines, we could potentially develop a lung-on-a-chip. So this is just one of the things that I did, and the team from UP Diliman Ateneo have collaborated with to just replicate, to test the equipment that we have in the lab and see if we could actually reproduce some sort of diagnostic device or organ-on-a-chip, in this case a lung-on-a-chip, and see if we could achieve the same resolution with all the devices or equipment that we currently have. And mind you, these are very cheap equipment. I mean, compare it to Weiss Institute. So hoping that we could actually collaborate more with different institutions, academic and research institutions, and even partner with industrial companies to further develop new diagnostic devices at a very affordable price. So this is one of the goals of why I did the Balik Scientist program, to actually help move the technology, but in a very affordable way. The next slide shows you basically one of the integrated rapid microfluidic device that have been developed by a researcher in the US and Germany. So here you can see a fully integrated rapid microfluidic device translated from a conventional 96-well ELISA kit. So ELISA kit has been one of the standard ways of determining reactions, looking at reactions or detecting active components. You can see here the conceptual illustration of the device. So you see here a 96-well microplate fixed in your PDMS micropillar, which is then attached to this, I believe, is a thermoplastic chip. And you can see the schematic of how the device could actually do ELISA procedure inside the microfluidic chip. So if we could do ELISA inside the microfluidic chip, this would make it more, that this proposed device could make it more affordable. So meaning, and also more controlled in terms of pumping samples and limiting reagents. So one of the ways that we could develop detection kits. So ELISA is a typical procedure used to detect certain compounds, certain mostly active molecules in it. So if we could do that in a microfluidic chips, then we could essentially translate ELISA in a detection kit using microfluidic technology. So this is one of the advantages of microfluidics technology, because it's very small. We could also do multiple detections in just one device. The next one is drug discovery and drug development platform. So here, I'm just showing you an example of a microfluidic platform for combinatorial drug screening on this particular study is on drug cancer biopsies. So what we can do with microfluidic devices is we could design multiple channels so that we could actually detect reactions to, let's say, a particular cell and try to screen as many compounds, as many molecules, as many solutions as we could to try to detect which one could actually, let's say, detect cancer or even kill cancer. So the advantage of microfluidic chip is that because it's relatively small and you could control the flow of the solutions, and you could also basically control the ambient conditions inside it. You could grow cells in it or even tissues in it, mimicking, let's say, cancer, for example. And then you could use that essentially tissue on a cell or even organ on a cell to develop or discover different drugs by trying to see the reaction of that particular tissue or cell or organ to your different active compounds. So this picture on the right is just to give you an idea of some in vivo validation that they did for drug combinations of, in this particular study, they used mouse as the models. But the advantage of microfluidics, at least for now, is that we're able to actually grow actual human cells and human tissues inside the microfluidic chip. So it also helps us move on from the typical animal models to actually developing organs on the chips based on humans, which is closer to closer than any animal model. So like I mentioned a while ago, this is an exit lecture. So I will be going through all the works that have been done as well during the BSP stint. So here I'm showing just a collaborative research proposals that we've managed to establish and completed. So a lot of it revolves around capacity building proposal for microfluidics device laboratories. So one of the things that I really envision is that we'll be able to develop microfluidic labs in various regions of the Philippines, because if we are able to do this, we could basically screen a lot of drugs or active compounds at the same time. So the Philippines has massive and very diverse fauna and flora that we could explore to detect different compounds that could be used as medicines. And we could screen as many compounds as we could in various locations with various fauna and flora. And establishing those microfluidics labs is a step forward to that. And so we also established, well, almost completed and ready for submission, some of the research and development applications, such as, like I mentioned, there are a lot of ASF viral infection a couple of months ago. So we're doing a proposal on this, and it's also ready for submission on top of the capacity building proposals for both UP Diliman and UP Alumna. Next is, I believe that it's always more efficient for academic research institutions to actually tie up or partner with industries or with other research facilities to, with the Philippines, we have very limited resources in terms of equipment, high-end equipment. And one way of achieving a really impactful science in an efficient and economical way is for us to collaborate with various universities, with various institutions, which may have the equipment that we need that we don't, but and at the same time forge expertise in various facilities or various academic institutions in the Philippines, and then tie up or partner with industries who may be able to provide us high-end equipment. So one of the things that I focus on during the stint is to develop or forge collaborations and partnerships with various professors from different universities, and also to link up with research facilities like biotechnology, UPLB, and the NICE at Ateneo. And I'm showing to you here two two figures, or basically, which is a product collaboration between Ateneo, UP, and Philippine Science High School. So we realized that during just basic, discussions and meeting with people who are also very keen on microfluidics technology, that we actually have equipment in various locations that we could utilize together to at least start doing manufacturing of microfluidics with a certain, of course, limited resolution, and eventually develop that into a more applicable or a highly targeted microfluidic platform for health research, for example. So here it's just showing you like the capacity that we have in terms of developing or laser cutting certain devices, and at the same time imaging, checking the microfluidic device if it's something that we could actually achieve with the limited resources that we have, while waiting to actually establish a microfluidics lab. Another, so I mentioned this a while ago, another thing that is quite relevant during the BSP is to also incorporate microfluidics technology in terms of research and also academic like courses or training, because this is not really a new technology in other parts of the world, but this is not yet incorporated in the Philippines in an extensive way that we would like, especially with the current situation. I think technology like this that could be translated and could be made affordable, portable, are much needed. So one way of doing that is to tie up with people or with industries, with facilities, and also to develop biotech startups for microfluidics technologies or biomedical devices like this, to basically hasten research development and device production. So apart from tying up with research facilities, we could work with industries, pharmaceutical companies who might benefit from microfluidics technology as well, in the hopes that through this technology, we could develop actual devices that are very much useful for the Philippines in terms of drug discovery and drug development, and eventually point of care testing. So that's the end goal, to be able to actually have devices that we could easily and affordably transport in different islands of the Philippines, and could be used not just to detect diseases, but to actually start developing new drugs, start screening new compounds, and producing our own medications. So here I'm just showing you other things that have been done during the BSP. So a lot of it, because of the pandemic, have been moved to webinar series. So I did a seminar on plant and chip. So this is where I discuss different technologies that we could utilize in the Philippines regarding plant extraction and screening in a more economical way using microfluidics technology. Another seminar focuses on organ on a chip, which is the future of drug discovery. So how we could actually build lung on a chip, brain on a chip, liver on a chip inside these microfluidic devices. And then the last seminar is in a more complex system, which is what we call systems on a chip. So basically, we can use this to transform drug development and disease modeling eventually. So those are the things that I did for the webinar series. I'd rather much prefer this on site so that you could also like practice, but then the pandemic gets in the way. But the idea is to actually introduce this sort of technology to our young researchers and young students who might be able to, extract all their creativity to produce a more advanced systems that could be used in the Philippines, like a more advanced yet affordable systems that could be used in the Philippines. And then with the BSP, we also did training courses. So I did a lot of training courses in terms of computational microfluidics. So the idea here is that we train more researchers and scientists to first do numerical simulations to develop designs using CAD in a microfluidics. So this makes it less expensive. So we don't want to produce a lot of microfluidic devices that we have to troubleshoot in the lab. So we wanted to see if we could actually get something that could be working numerically, and then we produced that. So the second training course, we did a lot of fabrication techniques and actually did a hands-on laboratory training on developing PDMS-based microfluidic at UP Diliman laboratory. Unfortunately, due to pandemic, we have limited the participants to 15, and we can only do like 2 groups at a time in the training. But I'm hoping that the people who have had this training could also train their students eventually. So this is one of the chips that we produce. This is the Langona chip, but at the same time, during this training course, we actually produce a more relevant designs and devices for the Philippines in terms of health and biomedical applications. And I'll show you that in the next slide. So during this training, I've asked the engineers and scientists in this course to actually develop 2 point of care testing, and that's dengue detection kits and TB detection kit or detection platform, because these are 2 highly relevant diseases in the Philippines. And as you can see, so this is one of the design that has been developed by group 1 by my students Nortes Ordillo and Ordonez. And this is just to show you that the Philippines, that Filipino minds are brilliant, and they could develop new designs for dengue detection kit that we could actually manufacture in the Philippines if we have the sufficient equipment to do it. And the relevant side of this is that we know that we could design and manufacture things that could be used and translated for health applications like dengue. Another design that I'm showing you here is by Scara and group. So this is a very simple design that this could be manufactured using laser cutting technology, and we could do this and test this in labs in UP Diliman and UP Los Baños, in fact. So this is just to show you that microfluidics technology is not a far-fetched technology. It's something that we could do in the Philippines, and it's something that we should take advantage of. So another applications that I asked my student to do is the TB point of care diagnostic tool. So this is just one of the tools that we developed in class. So this is by Sanglay, Lintal, and Santibanez. And so they developed a microfluidic design for the detection of TB, I think from urine. Another group developed something using blood sample, which is more complex and complicated. So these are the things that we could do in the Philippines if we have established microfluidics laboratories in academic institutions and even research institutions. So also on top of that, one of the visions or objectives of my BSP is to also incorporate microfluidics in Philippine education, not just the higher education institutions, but like University of the Philippines or Ateneo, etc., but also in high school courses. So one of the collaborative works that I've done is through the Philippine Science High School in the main campus and Bicol Region campus, wherein they use laser cutter and 3D printing to actually develop small features that could be also used as a mold for a microfluidic device. So we could incorporate this as a research projects for high school students. And also more relevantly, or at least the one that I started working on, is to incorporate this in course as a course in University of the Philippines. So not just as a thesis research for graduate level and undergraduate level, but also as an actual course, so either a seminar or an undergraduate course like 197, for example. So this could help with, this could introduce certain small technology or nanotechnology or microfluidics technology in the curriculum of students, and they could explore this as a good way of understanding actually fundamentals in engineering principles and fluid mechanics. So this course could also be incorporated there. And eventually, we would like to, of course, look into establishing Center for Drug Discovery and Development in an affordable way. And I think one step of doing that is to establish microfluidics laboratories in various institutions which have specific strength. So UPLB, for example, is very good with plant science, is excellent in plant science and agriculture, and has the capacity to do that with their chemical engineering department and their physics department. Also, UP Diliman, for example, has a very good physics and chemical engineering groups, and at the same time have access to biotech laboratories. So collaborations on two of those laboratories could help with, let's say, detections of more human applications, diagnostics, or point of care testing. While UP Los Baños have the veterinary medicine, so they could collaborate and work along with a lot of possible viruses that are affecting the livestock, for example. So all this future directions could be like, is motivated by actually developing a good and efficient way of maximizing the Philippines fauna and flora by using that, by exploiting that, so that we could actually benefit from it in terms of discovery and drug development. And eventually, I really hope that we'll be able to establish more biotech and medical startups in the Philippines and Southeast Asia. And this BSP stint that I have is actually geared towards that as well, to develop a name for the Philippines in terms of biomedical devices and medical startups that are affordable and portable. So with this, I end my exit lecture. I hope that gives you, I know it's a very brief lecture on point of care microfluidics, but hopefully you could also like watch the webinars and the training lectures that I've given in the past, so you'll have more idea on what the other applications of microfluidics are, and the in-depth examples of each of the microfluidic technology applied to plant, to organs, or to systems. And I'd like to thank UP Diliman, UP Los Baños, and PCHRD for giving me this opportunity to at least, in a way, contribute to the country. So salamat po, and I'm ready for questions. Sorry, medyo low bat na ako at 11 na dito.
That's a very informative presentation. A round of applause, please, for our Balik scientists. Thank you very much, Dr. Fojas, for sharing your knowledge and recommendations to us. Now we open the floor for questions from our audience. There's a question from Scott Berza. For the dengue microfluidic system, how much would a test kit cost? And in comparison to the NS1 immunochromatographic assay test kits?
So in terms of costs, I could not really give you any estimate on that. What I could give you is that relatively the microfluidic devices uses a very small amount, like nano to micro ml, actually mostly nano ml. So that would reduce the cost of reagents for quite significantly. And on top of that, the microfluidic devices could be developed using PDMS. So PDMS is quite cheap. And you could also use like plastics, so polyethylene base as your devices. So that's also relatively cheap. And if we are able to use equipment which is relatively affordable in the Philippines, like 3D printers and laser cutters, which have a lot of uses. So that reduces the cost. But estimating wise, I'm not sure in particular how much one detection kit could cost. It depends on the reagent that you have in there, the detection compound that you have in there.
The next question from Mario Domingo. Dr. Fojas, first, congratulations for your work. Among other things, how do we ensure sustainability of the work you've started in microfluidics in the country? You have provided very succinct recommendations for capacity building, but with all of the many things we are pursuing, how can we keep the focus on and sustain this stream of work?
So one of the things that could help us actually move this forward, apart from the establishment of microfluidics, is to tie up with industries and private partnerships, really, to develop bio startups on this, to link up with, let's say, pharmaceutical companies who might need us to develop high throughput drug screening microfluidic technology, or to actually build biomedical device companies wherein we explore the production of microfluidics for various health applications. So that's one goal, and those are two goals that we could actually do to move forward. And this is also one of the reasons why I'm keen on going back to the Philippines, because not only would I want the lab to be actually fully equipped and established, and to have as many engineers and scientists and researchers trained to do and manufacture microfluidics, but to also see through it that we can have marketable devices that actually could benefit the community. So I believe the DOST is doing a great job with moving forward a lot of research proposals. And I'm hoping that the DOH and DOST could actually have a more collaborative research work wherein research outputs like this, like devices, could be easily translated clinically. And yeah, so those are the things that I think could help us make it sustainable.
From an anonymous attendee, how efficient is the chip to help in growing human tissues?
It really depends on the human tissue. So my experience with the lab, so with cancer cells, it's really fast. With let's say lung on a chip, it's relatively easier compared to brain on a chip. So it depends on the tissue that you're growing and the type of cells that you're actually growing. So primary neurons, for example, is primary cortical neurons is more easy to grow than primary hippocampal neurons. So those things you have to so you couldn't really say how easy. It depends on a lot of parameters, and depends on your cells, and depends on what organ you're trying to build. For example, heart is more complicated. It's a muscle tissue that you're building, and at the same time you have vascularization. You need to have constant supply of blood in it, so it's more complex. Did I answer your question?
Next question from Richard Tambalo. Thank you very much for the information. Were you able to establish a collaboration with a privately owned entity or group interested for locally manufacturing versions of the technology?
So right now, all the talks that I had majority are in academic institutions, but at the moment, I am establishing a startup for microfluidics with other professors and private partners. So that's one goal of doing it, to hasten the creation, or rather development, of the microfluidic technology in the Philippines. Other partners could be pharmaceutical companies like Pasqua Laboratories. So I have an initial talk with 2 people from Pasqua Laboratories. So let's see how that goes too.
From Mr. Limuel Pertoliano, how efficient is microfluidics in culturing bacteria, virus, and animal cells? And can microfluidics chips be used in bionic limbs?
Microfluidics could definitely be used in bionic. How efficient it is, again, the same question, because the same answer to the previous question. In terms of, it depends on how you're controlling the condition, the ambient condition of your system. So bacteria, it's very easy. Virus, it depends, because with virus, you need a host, and it depends on what sort of cell you're going to use. So it's a multi-parameter system. So you have what microfluidic does is that it's because it's very tiny, you could easily control the different conditions inside the chip. So if you can control those conditions that are conducive for that particular cell or microorganism, then you can definitely emulate the environment at which they would grow.
From Mr. Glenn Samson, are these microfluidics kits have higher yield than the kits being used now? And for the TB detection kit using urine as specimen, what is the accuracy of this and what will be the timeline for this kit to be used in detection of TB?
So those are the initial designs that we developed during the training course that I did. So they haven't been tested. There is an initial manufacturing of the device using just laser cutter, but those are not in scale. So essentially, without a sufficient microfluidic laboratory, we won't be able to actually test the devices. Those are just initial designs. So there is no efficiency or selectivity or sensitivity for those devices yet. So it's yet to be tested and it's yet to be manufactured.
From Mr. Descardenas, happy with this that can be adopted locally, micro and fluid, a continuum. I am curious with the solvents used compartmentalized.
So it depends on the type of solvent. So one research that I did is that we're trying to have different reagent of solutions for testing how the neurons react to various compounds, and those compounds are highly tolerated by PDMS, for example. So you could use PDMS. With microfluidics, you can use other materials too. So it depends on your solvent. If your solvent could be tolerated by PDMS, in general, PDMS is used for biological applications. So you might want to try solutions that are not corrosive or could melt the PDMS. So, but then again, like I mentioned, microfluidic devices, you could develop them in various materials, not just PDMS.
Another question, what further steps are needed for the microfluidic devices designed by the trainees for dengue and TB be further developed?
So one way so they did numerical simulations for the designs that they did. And we also tried to produce it in the lab with the limited equipment that we have and with really troubleshoot equipment that we have. And a lot of improvised equipment that we have. So the next step would be to actually have a high resolution, either laser cutter, or you can do soft lithography and develop the device and start adding your detection compound or your receptor, depending on the type of detection method that you're using for TB, for example, and then do a lab testing for that. So with developing detection kits using microfluidics, there is like a feedback loop. So you design it numerically, you check if there is enough flow, you check the pressure gradient, if it's enough not to push your, let's say, active compounds out of your detection or cleave your detection layer, for example. You do that a lot with numerical simulations, and you develop the device, manufacture it, and test it in the lab and see if your numerical simulation actually corresponds to your experimental setup. And then, of course, you have to test that together with your standard detection. So if you're using for I think for TB we have is it Gen Amplify, which is similar to PCR. So I forgot the ones that is being used by the Philippines. We have a lot of those devices in the country. So you could use that as a standard and compare that with the microfluidics to see if you have enough sensitivity and selectivity to actually say that your microfluidic device for TB detection is at par with the current standard technology. And that's how you do it.
Thank you very much po. And that is all the time we have for the question and answer. We truly appreciate the knowledge and expertise that Dr. Fojas has unselfishly shared with us. But before we let Dr. Fojas leave, the DOST PCHRD would like to present the certificate to her. Please let me read. The Department of Science and Technology and the Philippine Council for Health Research and Development present this certificate of appreciation to Dr. Jhalique Jane R. Fojas in grateful acknowledgement and sincere appreciation for sharing her time, expertise, and valuable service under the Balik Scientist Program short-term category, given on the 6th day of July 2021 at the University of the Philippines, Diliman, Quezon City. Signed, Jaime C. Montoya, Executive Director 3, DOST PCHRD. Would you like to say a few words, Dr. Fojas?
Thank you so much to DOST PCHRD, especially to Ma'am Paula and Ma'am Laila. And thank you so much to UP, especially to Dr. De Leon and Sir Rex Demafilis. So I'm really grateful for this opportunity. And I thank the Filipino people. If it wasn't for your taxes, I wouldn't get the education that I have, and I wouldn't be able to do this Balik Scientist program. So salamat po.
Again, thank you, Dr. Fojas, for sharing your valuable knowledge and expertise with us. Another round of applause, please, for our speaker. Now, to deliver his closing remarks, we would like to hear from the organization that brought Dr. Fojas to the Philippines through the Balik Scientist program. Let us welcome the DOST PCHRD Executive Director, Dr. Jaime Montoya.
Let me first acknowledge the presence of our guests, UP Diliman Vice Chancellor Gonzalo Campo Amor II, Professor Rizalina De Leon, and of course, Dr. Jhalique Jane Fojas. Colleagues, ladies and gentlemen, good morning. On behalf of the Department of Science and Technology, Philippine Council for Health Research and Development, I would like to congratulate Dr. Jhalique Jane Fojas one of the DOST PCHRD Balik Scientist Program awardees, for completing her short-term engagement with the University of the Philippines Diliman and UP Los Baños. The Balik Scientist Act was passed to further encourage our foreign-based Filipino scientists, engineers, and health and medical experts to return to our country to share their knowledge and actively participate in the government's efforts in research and development, develop better innovations, and foster greater information exchange that will lead to progress and advancement for both the community and the academe. Since then, our efforts towards supporting more researchers have been institutionalized and has enabled us to generate more studies and technologies that are geared towards making the lives of Filipinos better. As such, we would like to thank you, Dr. Fojas, for your utmost service. We hope that the expertise you have shared, especially in the field of microfluidics and diagnostics, has provided significant contributions to the development of a more accurate and point-of-care detection for diseases. We are certain that your experiences sa Balik Scientists would inspire other kababayan scientists to join the Balik Scientist program. We wish you success in all your future endeavors, and we hope that you will continue working with our local scientists. We would also like to extend our sincerest appreciation to the University of the Philippines Diliman and University of the Philippines Los Baños for being Dr. Foja's second home during her stay here. I am sure that more than the learnings, you have made her stay both meaningful and memorable. Maraming salamat po at mabuhay po tayong lahat.
Thank you very much, Director Montoya, for your inspiring message to everyone in this webinar. Before we end, may we request for a photo opportunity to everyone here in Zoom together with our Balik scientist, Dr. Fojas. Please turn on your cameras po.
Pwede kayang yung nasa attendees. Promote lahat. Para makasama sila.
Pwede.
Pinopromote ko lang yung iba. Para marami. Okay. Patulong na lang please Ives tsaka Sir July. Konti na lang. Wala palang promote call mo.
Thank you. Thank you so much, Dr. Fojas. Thank you.
Thank you, Jal. Congratulations, Jal.
Salamat po.
Nakamute pala ako.
Thank you po,
Salamat
Miss
po.
Cherry. Thank you po,
Jal.
Miss Cherry.
Thank you po.
Dito na ba lahat? Na lang. I don't know if yung iba nasa ano. Pwede na? Okay. I think we're complete. Yung iba kasi hindi I think they have to accept. Okay.
Hindi pa po na-accept nung iba.
Oo nga eh. But yeah, I think we can go ahead with that.
3 pages po.
Okay.
Smile po muna. So,
Smile po.
okay. 1, 2, 3. Wait lang. Next page. Last one.
Keep smiling kasi hindi natin alam tayo ngayon.
Po. Thank you very much.
Thank you.
Thank you. Well, that brings us to the end of
Thank
this
you.
event. Thank you po.
Thank you po. Thank you po.
Salamat.
Thank you. Thank you,
This
Sir
Zoom
Red.
webinar has been brought to you by the University of the Philippines, University Computer Center, and the Department of Chemical Engineering in UP Diliman. Again, thank you for joining us and have a great week ahead. Thank you po.
Thank you,
Thank
Chair.
Yes
you.
po. Thank you po.
Ito po.
Thank you.
Congrats. Thank you, PCHRD. Ma'am Laila, thank you. Sir Rex, thank you.
Thank you po. Thank you, ma'am.
Congratulations.
Thank you, everyone.
Thank you po.